Hey everyone, today we are going to learn about controlling gene expression. We've already looked at transcription, translation, and DNA replication, but now we're going to look at when these processes are turned on and when they're turned off. And in particular, we're going to look at an example called the Lac Operon. And this is just a specific example of a group of genes that we're going to look at for when they get turned on and turned off and how they get turned on or turned off. So we're just going to write down a few kind of quick ideas about the whole concept of how we control gene expression to start off with. So the first important concept of this is that not all proteins need to be transcribed or translated at all times. So let's just write this down. Another way that we can say transcribed and translated, like these are two processes that are required for genes to be expressed. So we can also just call the combined process of transcription and translation as like gene expression. That's what we mean when we're talking about gene expression. So there definitely are some proteins that do need to be expressed at all times, and we call those housekeeping genes. So let's write that down as well. And some examples of proteins that we would constantly need are things like DNA polymerase or helicase are two examples that I can think of off the top of my head, because we are always going to need the proteins involved in DNA replication, like enzymes. And other examples are like things we need for metabolism, like the enzymes we need to metabolize energy, because we always need to be kind of like cycling ATP and energy. So I'm just going to write, for example, all the DNA polymerases are good examples. Helicase is another one used in replication that we need. So these types of proteins are regulated by housekeeping genes. Right, because a gene is just a segment of DNA, so we can describe proteins as being regulated by the gene expression, right? So when a certain gene gets expressed, we get a protein. If there's proteins that we need all the time, those proteins, their expression will be regulated by the genes called housekeeping genes, which makes sense. Like we think of housekeeping genes as just like the ones that are always around, kind of cleaning stuff up, making sure everything is running properly. And then we will look at, we'll jump into the Lac Operon now, and that's an example of a non-housekeeping gene. So the Lac Operon is actually just kind of like a collection of genes in the bacteria E. coli. So it's an example of prokaryotic gene expression, which we'll talk about why we're looking specifically at prokaryotes in a second. So an example of gene expression in E. coli is the regulation of lactose metabolism genes. There are 3 genes that are involved in this gene expression. Okay, so an example of gene expression in E. coli is the regulation of lactose metabolism genes. So lactose is something that can enter bacterial cells, and depending on whether that lactose is in the cell or not, the E. coli will regulate its gene expression of proteins that metabolize that lactose. That's similar to us, like for humans, we only express the genes for that code for insulin when we have glucose in our blood. So these 3 genes in E. coli need to be expressed in order for the bacteria to break down and metabolize the lactose in the cell. So that's why we call it the Lac Operon. So we're just going to draw a little diagram here to help explain this. So we'll draw 2 cells. And we'll draw some DNA in here. And then remembering that genes are just a chunk of DNA. These are going to be our 3 genes that need to be expressed in order to break down the lactose. So if we have 2 cells, and this first cell has these little molecules in it, which we will label as lactose. So if lactose is present, the cell needs those genes in the Lac Operon to be expressed. If there is no lactose, we don't need them expressed. And we're going to dive into how exactly these genes are regulated. Like, how does the cell even know that there's lactose there, or how does this impact how the genes are expressed? We're going to look exactly at how that works, but this is the main idea here. And to be even more specific, these different genes, these 3 genes, have different names. So the first kind of component of the Lac Operon is called Lac Z. The middle one is called Lac Y, Lac A. So again, these are just 3 different genes. All of these genes need to be expressed as part of the Lac Operon because each of them help break down lactose in a different way. We don't need to worry about what exactly they code for, like what proteins they produce and how that breaks down lactose. We just need to know that Lac Z, Lac Y, and Lac A are the genes that need to be expressed in order to break down lactose in this cell. So we're going to do the same thing here. And we'll draw some better diagrams in a second, but this is just the main idea. Also, let's just correct our grammar here, not expressed. Okay, so now we're going to dive a little bit deeper into the Lac Operon and understanding how these genes, these 3 genes that kind of make up the Lac Operon, are expressed. And in order to do that, we have to look further upstream on the DNA that these 3 genes are on and look at different areas, including the promoter, which we've already talked about in class, and then a few other different areas that are important for the regulation or expression of these 3 genes. So the first area that we can point out is upstream here. We have the promoter, and beside that, the operator. And these are just 2 different kind of chunks of DNA that are responsible for, in part, how we get the expression of these 3 Lac Operon genes. So let's make these different colors. We have our promoter and operator. And then a little bit further upstream, we have another gene. This is called Lac I. It's just another gene similar to the 3 that we have down here, except it performs a different role that is necessary for the expression of Lac Z, Lac Y, and Lac A, which is kind of neat. Like, just upstream of those genes, there's another gene that first needs to be expressed in order for expression of the next genes down the DNA to work out. And the way that Lac I works is if this undergoes transcription and then translation, we get something called a repressor. So we get this protein that we call a repressor. And this repressor is super important to the function of the Lac Operon. And this little kind of area here, this kind of like little indent, we'll label that too as this is going to be a binding site for what we're going to call the inducer, because this protein needs to be induced by something in order to work. It has this little special binding spot there, and that something is going to be the lactose. So if the lactose is present, it will be able to kind of bind onto the repressor and change whether or not the Lac Operon can be expressed. And then we'll just label the promoter here as being So we know that the promoter is where gene expression begins. That's where the RNA polymerase will come on and kind of attach to the DNA in order to transcribe it. And because it's bacterial, we actually know that this sequence is going to be in there. And then next we have the operator, which up here we will write controls transcription. The operator plays a huge role because the operator is where the repressor protein is going to bind to, and depending on whether that repressor is bound to the operator or not, transcription will occur or not occur for Lac Z, Lac Y, and Lac A. The reason that the operator and the promoter are so close together is because if this repressor is attached to the operator, then RNA polymerase is too big and it can't kind of attach or jump onto the promoter. It blocks the RNA polymerase from attaching to the promoter. So their location is important on this strand of DNA. So now we're going to look at a few different situations that can happen inside the cell of E. coli and then how that affects whether or not these 3 Lac genes of the Lac Operon are being expressed or not. So we'll say situation 1 is no lactose. So if lactose is absent, the repressor is expressed. So if lactose is absent, the repressor is expressed, and Lac Z, Y, and A genes are not expressed. So the repressor being expressed means, remember, this gets expressed by the Lac I gene. So let's draw our repressor here. So the Lac I codes for the repressor. Great. We have our repressor. And the way that this repressor works is it kind of travels on and latches on to the operator. So we can show that, comes on over here and latches on there. And what this does is it blocks RNA polymerase from being able to get by. So if this is our big RNA polymerase, it can't get by. So these genes can't get expressed because the RNA polymerase can't get past the operator because the repressor is there. So we can label these things. And this makes sense because if there isn't any lactose in the cell, then we don't need those Lac Z, Y, and A genes transcribed or translated, because the cell has no lactose that it needs to metabolize. Now we'll look at if lactose is present. Okay, so if lactose is present, the repressor is still expressed by Lac I, so we can just draw this in again. We know for sure that this repressor is still going to be expressed. So we still get a repressor. However, if there's lactose in the cell, so we're going to draw lactose as these little diamond shapes. These are little lactose molecules. And they happen to fit perfectly into this binding site of the repressor. So if lactose is present, this repressor now cannot bind to the operator, because now that it has that inducer in it, the lactose, it changes the shape of the enzyme. You guys know how when enzymes are acting as like the active site for a different type of like protein, then it can change shape, and now this lactose being here no longer allows the repressor to bind to the operator. And we call lactose in this case the inducer, so let's label these. So, since the repressor can no longer bind to the operator, our RNA polymerase is free to move along here and transcribe and translate Lac Y, Lac Z, and Lac A. So these are all expressed, and now they're expressed to create the proteins that the cell needs to metabolize the lactose that is in the cell. I'll label this again here too.